Hey guys, today I'm going to walk you through some notes over protein synthesis. This is the next step in our process of DNA replication. So here we are, we've already talked about DNA replication, so let's review that really quickly. This has to occur before the cell can divide. It produces two identical double helix DNA molecules. So remember, we go from one strand of DNA and it gets split into two. This happens in two phases. Step 1, DNA helicase unzips the DNA at the replication fork. Step 2, DNA polymerase actually attaches the complementary nucleotides to the new strand. Okay? And then there are special enzymes that proofread to make sure there's not any mistakes within that strand. All right, so let's take this strand of DNA here. This is going to be problem number 1 in your notes. Go ahead and replicate it. Give it the complementary strand. Okay, so it should look something like that. TAC converts into ATG, TTG into AAC, and so on and so forth down the line. You're just making the complementary sequence there, right, when we replicate. So that's kind of the review of replication. Today we're going to be going to the next step, all right? The ultimate goal in this process is to go from DNA to proteins, okay? You're turning in the blueprint that is DNA. You're actually using that to make proteins that will be used throughout the body in different ways. So DNA and RNA are both involved in the production of proteins, okay? Here's a bit of a chart. DNA contains the sugar deoxyribose. RNA contains the sugar ribose. That's the D in DNA and the R in RNA. In DNA, there are 2 strands. In RNA, there are only 1. The bases for DNA are A, C, G, and T. In RNA, the T gets replaced with a U. It's A, C, G, and U. So anytime you would have a T in your genetic sequence, you would actually put a U for RNA. So A would pair with U in RNA instead of A with T, as in DNA. All right, so the next step, we've replicated our DNA. Now it's time to transcribe it. All right, the steps to making proteins are called transcription and translation. This whole process together is protein synthesis, the making of a protein. Okay, we start with DNA. That DNA gets converted into mRNA. That mRNA is then used to make proteins. So transcription is information coded in DNA actually gets transferred to RNA. This happens in the nucleus of our cells. This starts with RNA polymerase binding to a promoter that's a sequence on our DNA strand, and it actually unwinds the DNA just like DNA helicase does, but this is RNA polymerase. Okay, the promoter is an area within the code with lots of A's and T's. And then the separated DNA, as it's unwound, serves as the template for the mRNA strand. Okay, RNA polymerase then moves along the nucleotides of DNA and adds the complementary RNA bases. So it takes a strand of DNA and makes a new strand, of RNA from that strand. Okay, C still pairs with G, but now A pairs with U instead of T. Once the RNA strand is complete, it's now called mRNA. That's messenger RNA. mRNA leaves the nucleus and then enters the cytoplasm. It's actually carrying a message. It's the messenger RNA. So it leaves the nucleus and enters the cytoplasm. The DNA then rewinds itself back to normal, goes on about its day. Okay, exons and introns. Okay, sometimes this message has extra stuff in it, and we have to cut that out when our DNA gets converted into RNA. These are things that are called exons and introns. They are specific nucleotide sequences in the genetic code that are involved in producing proteins. Exons are bases that are transcribed into mRNA and will eventually code for proteins. Introns are found in between the exons, but they're not transcribed. They're like space fillers. So when you look at this picture here, we have some introns and exons within our little strand of DNA. Okay, well, when we make the RNA strand, we cut out the introns, and then we're left with only the exons together. Okay, like I said, you're taking out the excess kind of out of the genetic code. You're kind of purifying it for the mRNA strand. Okay, exons are the part you keep, introns are the part that you actually cut out. So Now let's practice transcription. We're going to take our DNA sequence and we're going to turn it into mRNA. So TAC, TTG, GGG, on down the line, those will now get converted into mRNA. So we'll write the complementary RNA bases. So it should look something like this. Now TAC becomes AUG, right? Instead of having a TG, it gets replaced with a U, so it's AUG. T still turns and bonds with A, so it's AUG, AAC, CCC, on down the line there till you have those 6 parts. So, like I said, the steps are here. Okay, we've talked about DNA, we've talked about mRNA. Now let's talk about making proteins. This is the next step. This is the last step in the process. It's called translation. So we have DNA replication, that's copying itself. DNA transcription, that's it being turned into another language, essentially DNA to RNA. And then translation, that RNA is being turned into an amino acid. So translation, we take 3-letter RNA sequences and translate those into amino acids. Amino acids are the building blocks for proteins. And remember, proteins are used throughout the body for lots and lots of things. Okay? This all happens in the ribosome, which is in the cytoplasm of the cell. So, a couple phrases. If you're in Spanish, French, or German, you might be familiar with these. These are standard greetings in different languages. So, como se llama, como tu te pel, and auf Wiedersehen. Okay? When you change something from one language to another, this is called translating, right? Como se llama, I think it means how are you doing. Auf Wiedersehen is goodbye. I don't know the French one, but that's okay. You understand what I'm saying here. You're taking this from Spanish, from French, and from German and putting it into English. That's actually translation. It works very similar within us. Ribosomes and tRNA, transfer RNA, take the strands of mRNA bases and turn them into amino acids, which will build into proteins. How this happens is each 3-letter segment of the mRNA is actually called a codon. Okay, so we have our strand of mRNA here, and you break it up by 3, and you have AUG and AAC. Those are 2 codons, those 3-letter sequences. Each codon specifies a particular amino acid. This is going to be used to be placed in a protein chain or a polypeptide chain. And I'll show you a different chart here in the future that's a little bit easier to read than this one, but there's one of the types of charts. tRNA actually acts as the translator because it transfers the amino acids to the mRNA at the ribosome. So it's this T-shaped molecule, and it actually attaches to the mRNA strand. Okay, and it has 2 binding sites. Binding site 1 on the mRNA strand, it binds to the mRNA strand, the codon. On the tRNA, that's called the anticodon, the opposite of the codon. Okay, and then on the other side is the specific amino acid that relates to that codon. So it carries the anticodon on one end and that specific amino acid on the other. One by one, the amino acids are brought over by the tRNA and kind of placed in this chain, right? So eventually you end up with this long chain of amino acids, which makes up a protein. So this one already has the answers filled in for you, but you can see what we've done here. So we have our DNA sequence in the first line. In the purplish pink lavender thing is our mRNA strand from earlier. Okay, we've now taken our mRNA strand and translated it into tRNA, right? These are the anticodons. If AUG is our codon, TAC is our anticodon. AAC is our codon, UUG is our anticodon. So like I said, it's the same process. Still no U's. I've actually made a mistake. This very first one, this THC, should actually be UAC. So that was my fault. So there's a mistake that I caught. But like I said, everything... there's another mistake. Nope, I lied. That's not a mistake. Just the one at the start. It should be UAC as the anticodon to start. Everything else looks right. Okay, so looking at this sentence, where do you start and where do you stop reading? Right? Mr. Anderson decided to give a pop quiz about Watson and Crick on Wednesday. How do you know where to start and how do you know where to stop? Okay? We know how to start and stop sentences based on the capital letter at the beginning and the period, question mark, whatever, punctuation at the end of that sentence, right? And we read from left to right, top to bottom. How does tRNA know where to start and where to stop reading on mRNA? There's always an amino acid chain that starts with AUG. Okay? This is always going to be the first time that it reads. It's looking down the line of this RNA sequence until it finds AUG. AUG is what we call the start codon. From there, it then adds bases until it reaches a stop codon, UAG, UAA, or UGA. You don't have to know the stop codons, but you definitely have to know the start codon. AUG is one that you will need to know. Okay, but don't worry about the stop codons. You just need to understand that there is a start and there is a stop. Okay, finally, after adding all of these bases in between the start codons and the stop codons, you've linked all these amino acids together, you've actually made a protein. What holds these amino acids together, it's called a peptide bond. Peptide is a fancy word for protein. It's a protein bond. It's a bond that only shows up in protein molecules, and it's known as a peptide bond. And then here we have our amino acid wheel. And you're going to use this here on the next slide, but I want to show you how it works first. So what you do, let's say you have AUG is our start codon. You'll start here in the middle with the biggest letters. You'll go A, then the second tier, U, G, methionine. Right? Do you see how I did that? AUG. First letter in the middle, second letter in the second wheel, and then the third one, you have these four choices. We did A, U, G, methionine. Okay, if I gave you another one, say CCC, you would start here. You go C, C, C. That gives you the amino acid proline. Okay. So here's another one I've already done for you. The DNA sequence we have right there at the top, we have our mRNA and we have our tRNA. Okay? From the mRNA strand, we get the amino acids, not the tRNA strand. The tRNA strands only carry them. Those are the anticodons. We translate the codons, not the anticodons. So you're translating the mRNA strand. So AUG from our wheel gives us the start codon. It's methionine. That's the amino acid. AAC gives us asparagine. CCC gives us proline. UCA gives us serine. GCU gives us alanine. And GGU gives us glycine. So that's kind of the run through here with protein synthesis, right? We've talked about DNA. We've talked about transcription and translation. Okay, now I want you to put it into practice. On today's Canvas page, there is a transcription and translation worksheet for you to practice on. Finish that and get it submitted, okay? Try not to have homework today. If you need to go back and rewatch the video, do that, all right? This is here to help you. So make sure that you watch the video and that you look over all of this stuff. And if you have questions, email me, come see me on Thursday. We'll definitely talk about this in class when we're together again on Friday. So be ready for that as well, all right? Good luck, and I'll see you guys soon.